We are honored to have Brian Diaz here from Emory. He is an assistant professor at Yerkes and the Department of Psychiatry. Brian grew up in India. He did his MSc there at the Tata Institute in Mumbai, correct? Studying mechanisms of depression in rodents. He then went on to do his PhD at the University of Texas, Austin, studying sexual behavior in lizards. As they do in Austin, I guess. He then got interested in the genetic mechanisms of behavior, switched species again to study fruit flies at Genelia Farms, and then he came to Emory to work with Kerry Ressler to work on the mechanisms of fear memory, where he had his remarkable discovery that I think he's going to tell us about today about transgenerational inheritance of fear memory in rodents. He is a great speaker. He's also a member of the Emory Tibet Neuroscience Initiative, where he goes to talk to Buddhist monks about neuroscience and mind matters and brain matters. And today he's going to tell us not about that, but about the mechanisms of memory inheritance. So, Brian, thank you.
So good morning, everyone, and I'd like to thank Chris and Bilal for the invitation to come and talk to you today. It's always nice to be able to come to a place where you hear so many things about, and I mean, this is a shameless plug for any potential collaborations that could come from this talk. This is a plea for help as well with some of the data that I'm going to show you, and so if light bulbs go off when we're talking about some of the big data things that we're doing, please do reach out, and I hope to reach out to you during the course of the next few months. Talks go pretty quickly, and so what I want to start off by doing is talk to you about the people who actually do the work and give them all the credit. All the criticism you throw my way, all the credit is to them. A lot of the work that I'm going to be talking about today is the work of two research specialists in the lab, Haj Awed and Soma Sanigrahi. And then Sarah Hunter is an undergraduate in the lab who's really taking us in different directions with the work that I'm going to be talking to you about, but I'm not going to be able to share too much of her work this morning. In his work on children, the Lebanese-American poet Khalil Gibran writes, "And a woman who held a babe against her bosom said, 'Speak to us of children.' And he said, 'Your children are not your children. They're the sons and daughters of life's longing for itself. You may give them your love, but not your thoughts, for they have their own thoughts. You may strive to be like them, but seek not to make them like you. You may give them your love, but not your thoughts, for they have their own thoughts.'" Historical events, data collected in the aftermath of those events, and modern neuroscience prove this to be completely false. Take, for example, Rachel Yehuda's work. Rachel's shown that babies who were in utero at the time that their mums directly experienced the 9/11 terrorist attacks had lower cortisol levels at age 1 compared to a comparable cohort of babies whose mothers did not directly experience those attacks. Rachel's group has also shown that lower cortisol levels are predictive of post-traumatic stress disorder developing in descendants of Holocaust survivors, even if those descendants did not experience the Holocaust themselves. Closer to home, there's work by Tanya Jovanovic, which has shown that children born to mothers who were abused, and even if this abuse happened before those children were conceived, those children brought into the laboratory present with higher anxiety and depression-like metrics compared to children who were not abused. And then, of course, we know across history, starting with the Dutch hunger winter in 1944, when food and ration was embargoed going into the Netherlands, and then a few famines that have occurred in Sweden and all the data collected in the aftermath of those famines, that nutritional impoverishment has profound imprints on descendant biology in how they process their food, mental health, and a suite of other metabolic disorders. So if you think that this happens only in humans, that's not true. And one of my favorite examples is this study with field crickets and wolf spiders. So you have gravid field crickets who are pregnant, and those field crickets were raised in two environments: high predation by wolf spiders or low predation by wolf spiders. And what these authors found is that the crickets who were born in an environment that had high predation by wolf spiders showed defensive behaviors to the wolf spider odor and did not approach that wolf spider odor. And so descendants bear profound imprints of ancestral environments, and ancestors transfer copious amounts of information to descendants. This happens across life stages, and this happens across species. The question then becomes, how do descendants inherit information from ancestors? And how is descendant neurobiology impacted by ancestral environments? To answer these questions, there have been a lot of animal models that are allowing us to be able to decipher how descendants bear imprints of ancestral environments. So take, for example, in the nutritional space, I'm showing you two papers here, one by Margaret Morris's group and the other by Ollie Rando's group, and both of these have a similar flavor. They took male rats, female rats, they manipulated their diets, and then they looked at the offspring, and they show that the offspring bear imprints of these altered diets, such that the offspring process their food differently. There's higher metrics of diabetes and a bunch of other metabolic symptoms. And so when it came to nutrition, we already knew that you could model this in the laboratory. When we were starting out these experiments that I'm going to be talking to you about, there was less known from a neuropsychiatric perspective. Shown here are two studies, the first by David Cruz, who I coincidentally did my PhD with. David took Michael Skinner's model, and what Skinner's model is they take female rats when they're pregnant, and they feed them a fungicide called vinclozolin. And what Michael and his group, and David in this paper showed, is that the F3 generation, remember it was only the F0 generation that was fed the vinclozolin, the F3 generation showed transgenerational imprints of this particular ancestral fungicide exposure. And in this case, they looked at mate preference. Michael Skinner's group has gone on to show that this occurs across several generations, and I think they're at F8 right now. Another example that I like to share is work by David Dietz and Eric Nestler's group, where you take a male mouse and let him get beat up upon by another male mouse. That's called social defeat. And when that male mouse who's been defeated goes on to sire offspring, they found that the offspring show higher depression and anxiety-like metrics in the lab. And so these examples show that you can inherit information, or at least transfer information, about acquired traits across generations. And there's been a lot of work, and I'll talk about this towards the end of my talk, from Isabel Mansui, Tracy Bale, and a suite of others, trying to get us to how this is actually happening. All of this is fine and dandy, but I think there's still gaps that persist. And those gaps come in two flavors. One is, can we prevent or reverse the effects of ancestral environments in descendants? And how is this actually happening? And I'm going to submit that to answer both of these questions, we need to be able to physically follow, in some shape or form, a cue across generations. We need to physically follow an ancestral experience that occurs in one generation across multiple generations. That, to me, is akin to looking for a small needle in a giant haystack. Where do you look for the imprints of the Holocaust? Where do you look for imprints of social defeat? Where do you look for imprints of fungicide exposure? That's where olfaction comes in. So the olfactory system, as is known to most of you in the room, you and I smell just like rodents smell. Within our nose, there are these neurons called olfactory sensory neurons in the olfactory epithelium. I'm going to call those OSNs, olfactory sensory neurons. OSNs send their axons into the brain through the cribriform plate, and they coalesce into functional units in the olfactory bulb called glomeruli. You can have millions of olfactory sensory neurons in your nose, but each sensory neuron will only express one of 1,000 different odorant receptors. Odorant receptors being the locks that bind all the odors in your environment. So an olfactory sensory neuron here, ORA, or OSN, is expressing ORA or red. This OSN is expressing yellow, blue, green, and purple. Why is that important? Spread across the nose, you're going to have a bunch of OSNs that express red. But no matter where they are in the nose, they're going to send their axons, and those axons are going to coalesce at the same place in the olfactory bulb into a functional unit called a glomerulus. The OSNs expressing green, all of them are going to go into a particular place in the olfactory bulb, again called a glomerulus. So there's an exquisite structural organization where you can visualize information going into the brain in the olfactory system. Why is this important? This is important because in the lab, we have mice where you can visualize specific OSN populations that express specific odorant receptors. So here you have the M71 lag-Z mouse. And what the M71 lag-Z mouse allows you to do is it allows you to stain blue all the sensory neurons in the nose that express the M71 receptor, and those send their axons into the brain, coalescing into the olfactory bulb. Why is this important? This is important because we know that there's an odor called acetophenone which goes and activates the M71 odorant receptor. We also know that an odorant propanol does not activate this receptor. And so now not only do you have a structural organization to the olfactory system, but you also have some kind of functional specificity. You can take this information, and what you can do is our premise was that you could take this information and condition these animals with an odor. So odor, shock, odor, shock, odor, shock, acetophenone plus shock, acetophenone plus shock, or propanol plus shock. The animal's going to become fearful of that odor. You then have a behavioral protocol in which a specific cue has taken on a salience and a valence. The olfactory system already has this functional and structural specificity in this animal. And you have a genetic locus, the M71 lag-Z locus in the acetophenone responsive olfactory sensory neurons, where you can look for imprints of this particular olfactory experience across generations. So remember, I said one of the things that I think we need to do to be able to understand how descendants inherit information about ancestral environments is actually follow that information. Here you can follow that information at the level of structure, visualize it in the next generation. You can follow it at the level of function because you can look at how those next generation respond to this particular odor. And maybe, just maybe, the imprints of this experience you would find in the sperm or the eggs of these animals, which allows it to go to the next generation. Everyone with me? I'd be happy to take any questions throughout the course of my talk if I don't want to lose anyone. All right, so this was where we thought we could go from an intergenerational perspective. What do we start with? So before I got to Kerry's lab, Seth Jones, who was a graduate student, put out a paper in 2008 where they took animals and fear conditioned them to acetophenone or propanol. ACE plus shock, or propanol plus shock, propanol plus shock. Shown here on the y-axis is something called fear potentiated startle. If you do that, some of you may have startled, some of you may have not. You put an animal in a really small environment and the animal startles. That's called startle response. If you pair that noise burst with a cue that the animal has been conditioned with, the animal is going to be more fearful and show a bigger response. And so what's shown on the y-axis is fear potentiated startle, a measure of fear. You have animals who were conditioned to acetophenone showing a high startle to acetophenone, but not to propanol. Animals who were conditioned propanol plus shock, show a high fear to propanol, but not to acetophenone. So there's a specificity of response when the animal is conditioned with a particular odor. Complementing this specificity was a neuroanatomical effect. such that in animals who were untrained, these are what the glomerulus looks like of an M71 lag Z animal who's not trained to any odor or just trained with propanol. When the animal is trained to acetophenone plus shock, there are more sensory neurons that express this particular odorant receptor that now send more axons into the brain. More axons into the brain means a larger glomerulus. And so there's an enhanced representation for that particular odorant receptor in the trained nose and in the trained olfactory bulb. So now the question becomes, what if we conditioned animals, we waited for some time, allowed for them to mate, got their offspring? How do their offspring respond to this particular odor? How does the olfactory system look like in these offspring? And what are the signatures of this olfactory experience in the sperm of these animals who'd been conditioned, or the eggs that have been conditioned? So here's what the behavioral data look like. Shown here is something called odor potentiated startle, OPS. It's not fear potentiated startle because we are not training these animals. These are F1s whose fathers had undergone some kind of odor experience or not, and then we're just testing their baseline response to these odors. When animals are trained to acetophenone, and then we get their offspring, so these are F1 ACE offspring, they show a higher startle to acetophenone compared to animals whose fathers were conditioned to propanol. And there's a specificity of response because when animals were conditioned to propanol, they sire offspring that are now more sensitive to propanol, but animals who were conditioned to acetophenone, their offspring are not showing a higher potentiated startle to propanol. When this work came out, a lot of press picked it up and said mice inherit the fear of their fathers. I'm here to stand before you today, to get some good food, which I've sequestered away because everyone told me I needed to. Now, I'm here today to tell you that it's not a fear response. You will never hear me say it's a fear response. It is a sensitivity to that particular odor. Their noses are set up to respond to lower concentrations of the odor, to just tell the animal, I'm anthropomorphizing here, something's happening, be alert. And we know that because we did a dose response, a concentration curve, and animals do indeed are able to detect lower concentrations of these particular odors based on what the F0s were conditioned to. So this is similar to you being able to detect the cologne or the perfume of someone you're familiar with 10 feet away, as opposed to them having to come really close to me for me to even detect what perfume or cologne that they're wearing. You're able to detect lower concentrations of that odor. So it's a sensitivity, not a fear. And we can talk about how we figure out whether it's a fear response or whether it's appetitive towards the end of the talk. So we have this enhanced sensitivity in the next generation. What does the neuroanatomy look like? So when you look at F1 home animals, so these are animals who are sired by individual or F0 males who are just hanging out in their home cage. That's what the glomeruli look like. When you have F0s conditioned to propanol, their offspring glomeruli look this way, no real change. But now in animals who've had no prior exposure to acetophenone, but their parents had been conditioned to acetophenone, this is what their glomeruli looks like. There's an enhanced representation for this particular odorant at baseline. And so not only was there an increased behavioral sensitivity to the odor, there was an enhanced olfactory representation for this odor. At this point, we asked the question, how is this happening? And it seemed like there were two possibilities. You could either have social transmission of information, which is very akin to cultural transmission, grandmothers or mothers telling their offspring about what the Holocaust was about, or there could be something within the sperm or egg that allowed for an inheritance of information to occur. We wanted to tease these possibilities apart. So we did 3 sets of experiments. I'm not going to have time to go into the details of them. But basically, all of these experiments suggested that there was an inheritance of our phenotypes, not a biological transmission, not a social transmission. So for example, we did in vitro fertilization. So the idea being, okay, maybe we've conditioned these males, maybe they've mated with these females and transferred some information about that conditioning to offspring. So what if we just take sperm from these males, do an IVF experiment in a dish, implant those into surrogates, will we still get those effects? And we did. So that implied to us that there was an inheritance of information, not so much a biological transmission. So the question is, how is this happening? BLUE is blue for an M71 LAC-Z sensory neuron in the F0 generation, and BLUE is blue for an M71 LAC-Z neuron in the F1 generation. What we thought was happening is you're making a lot of blue, you're making a little blue, a lot of blue, a little blue. And so we turned to epigenetics, the ability to read how the genetic code, how much of a gene is expressed versus not. And this is an embarrassingly simple cartoon of epigenetics, but it shows you 2 flavors. One is DNA methylation, which I know a lot of people in the room study. So you have methyl groups that are attached to CPG sites in the genome. We now know that methyl groups are not only the purview of CPG sites, and that's a conversation for a different time. And then you have histones around the DNA woven around histones, which tell a particular locus, all right, come and transcribe me, or if there's another set of histones, it's going to say, no, I'm not available to be transcribed. And so what you're doing is you're allowing for a particular genetic locus to either be transcribed or not. So if this information went through the sperm, which our IVF experiment suggested, then we would have to have some kind of clues as to what's going on in the sperm. And so we looked at histones at the M71 locus in the sperm of animals that have been conditioned with acetophenone or propanol and saw absolutely nothing. So at that point, we sent this off to Active Motif to do bisulfite sequencing. We now do this in the lab. At that time, we didn't have the ability, and so we sent it to Active Motif. And what I'm showing you here is percent methylation on the y-axis. And you can see that in the F0 ACE sperm, so these are animals who've been conditioned to acetophenone, there's a modest but significant decrease in methylation compared to sperm from animals who had been conditioned to propanol. This was not on every olfactory odorant receptor that we looked at, because when we look at OLFR6, which is not activated by acetophenone, we don't see this difference in methylation. I am not here to say that DNA methylation is what is giving us this causal mechanism that allows for the inheritance of our phenotypes across generations. And I'll tell you what I think it is towards the latter half of my talk. But what I'm here to say is that we think that DNA methylation gives us a readout that something salient happened to the animal and the sperm is registering that information. If you think that's far-fetched, I'm only going to draw your attention to more data coming out from fathers who are of an advanced paternal age, and we know that a lot of the birth issues that we might be seeing is because of DNA methylation marks being accrued or stripped away in the sperm of those fathers. And so DNA methylation in the sperm is a pretty big deal. But we're using it as a readout of saying that there's an olfactory experience, the sperm is registering it in some shape or form. Whether this is causal or not, I think is a topic for another conversation, and I think some of the technology needs to catch up. So when we sent this paper out, we showed that these effects went into the F2 generation as well, which the reviewers thought to tell us, so now show us that this methylation is also present in the F1 sperm. And so we went ahead and did that experiment, and we see a decrease in methylation even in the F1 sperm of animals whose F0s had been conditioned to acetophenone. So what I've shown you thus far is before I came into the lab, you could take an odor and shock an animal, and that animal then showed a fear towards that odor and then had an enhanced representation for that odor in its brain. We were able to show that this resulted in a decreased methylation in the sperm of these animals at that particular odorant receptor locus. When you trained the next generation, when you exposed the next generation to the odor, they had a higher startle to that particular odor and an enhanced representation for that odor in the brain, and this was at baseline. And we also showed that these effects persisted into the sperm of the F1s, giving us an effect in the F2 generation. I have this MOR23 GFP with Lyral here to remind myself to tell you, we also have another mouse in the lab, which is the MOR23 GFP mouse, where MOR23 neurons can be stained with GFP or express GFP. Lyral is an odor that targets those, and we see all of these effects even with that particular odor-OR pairing. And so this is not something specific to acetophenone. You can see this with different odors. So at this point, you should be saying, what? So I want you to draw your attention to two experiments. One is this beautiful work by Joe Todrank and Diego Restrepo, while working with Diego Restrepo, where what they did was they fed mum a particular diet, either acetophenone or mint, and we know that these particular odors will activate one odorant receptor versus another. And they did that at different points of gestation. And what they showed is that there was an enhanced representation, an increased glomerulus, where in the offspring for that particular odorant receptor when mum was fed that diet at different points during gestation. Almost identical to the work that we published, Ziv Williams put out a paper using fruit flies, where you can condition fruit flies and you see the same olfactory conditioned fruit flies, and you see the same effects in the F1 generation and the F2 generation as we saw. But Ziv, with the power of Drosophila genetics, was able to go and turn on and off mushroom body neurons and show where that particular olfactory processing was happening and how he could reverse those effects or prevent those sensitivities from going through one generation and then to the next generation. So we don't think this is something specific to mammals. In fact, we know a lot from the honeybee literature that this kind of stuff happens all the time, depending on foraging availability in terms of what the odorant bouquet is available to honeybees. And now we know that it's not specific to mammals because we've seen this in flies as well. We can talk about whether we think this is generalizable to other sensory modalities towards the end of the talk. So the question now becomes, all right, so this has been shown not only by us, by other people as well, and there's work by Regina Sullivan who's done things with odors and intergenerationally as well. The question is, what do you do with this information? And that hit home to me when we're talking to retired General Pete Chiarelli, whose question was, so what would you like me to do? Not enlist anyone in the US Army whose parents or grandparents suffer from PTSD or anxiety because of their own deployments or because of their own mental health issues? And so this is where these kinds of conversations take on a different meaning. And so on our end, what we need to ask ourselves is, can we reverse these effects? And these are the gaps that I was talking to you about. So the first gap was prevention or reversal, and the second was mechanism. Can we reverse these effects? Because if you can reverse these effects, maybe we can buffer future generations from the detrimental effects of ancestral traumas, or we can break cycles of inheritance. So can we? We turn to extinction training in animals to try and tease this apart. What extinction training is you have several groups. You have a home cage group. You have conditioned with acetophenone, which is what I've been talking about, ACE plus shock, ACE plus shock. You have exposure to acetophenone, which is just ACE, 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 ACE. But then you have, this is the money group, conditioned with acetophenone, exposed to acetophenone with no shock. And so what that means is ACE plus shock, ACE plus shock, ACE plus shock. So the animal becomes fearful of that odor, and then you extinguish that fear response by doing ACE, 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 ACE. And so the animal's fear response will diminish over time, or that's what we thought would happen. So Philomene Morrison, when she was a graduate student with Kerry, put out this paper where she took what we would consider F0 animals and she trained them to fear acetophenone, and then she subjected them to extinction training. So what Philomen is showing you on this graph is the percent freezing. So you can see when animals are trained to acetophenone, you have a high fear response. And then when you train them to acetophenone but subject them to extinction training, that fear response decreases over time. What Philomen was also able to show is that there's an enhanced representation for the M71 LAC-Z neurons 6 weeks post-conditioning, but after you extinguish those animals, after you perform extinction training, that enhanced representation comes back down to baseline. So there does seem to be a malleability within the F0 generation. The question then becomes, can we reverse effects in the F1 generation after extinction training the F0s? Everyone with me? Okay. So here's what the data looks like. The answer is yes, and we're trying to publish these data right now. When you take animals who've been conditioned to acetophenone, their offspring show a higher sensitivity to acetophenone, just like we showed earlier. But when you train animals to acetophenone and then extinguish their responses, you see that that sensitivity decreases. And that is complemented by the enhanced representation for the glomerulus seen in the F1 ACE animals, which then goes back down to baseline when you've trained F0s and then extinction train them. So there is, again, a malleability to this. We just don't know how this is happening. But remember I told you we looked at the sperm to see if they register information about all of this olfactory experience, and so we can partially rescue in the sperm the methylation marks. We see a decrease in methylation at the M71 LAC-Z locus in the F0 sperm, and when we extinguish those animals with acetophenone, we see a reversal towards baseline. I'm not going to say that it reverses it completely, and we can show this with Lyral as well. So why does this matter? This matters because way back when G by E was talked about a lot, it's still talked about a lot, gene by environment interactions. Epigenetics serves as that bridge that allows us to understand how environments might influence gene expression to give us a particular phenotype. And we know that this allows for a lot of developmental origins of health and disease that allow for one individual to go down a risk scenario, the other individual to go down a resilience scenario based on their environmental experiences. But I think what these data also show is that not only can learned sensitivities be propagated across generations, but also their reversal. So there is a malleability which we need to be able to tap into, because otherwise, for the most part, we'd be pretty screwed if every environmental experience had repercussions in the next generation. And for the most part, we're not. And so there is this malleability. The question is, what is the logic of that malleability, and how is that happening? So what I've shown you thus far is that in an F0 generation, you could take an odor and a foot shock. You could make that animal fearful of that odor and have an enhanced representation for that odor in the brain. And Philomen was able to show that you could reverse that with extinction training. What we were able to show with this intergenerational experiment is that via sperm-mediated inheritance of information, IVF-derived F1 and F2 offspring have an enhanced representation for odor and a behavioral sensitivity for that odor, which we can reverse unpublished data with extinction training. So there you have a scenario now where we followed that information, and that genetic haystack got a lot smaller, olfactory genes, and that needle got a lot bigger, which was that odorant receptor or odorant receptors. But not so quick. How is this happening? And to address how is this happening, you got to get at what are the critiques of this particular field. So I found the best way to address the critiques is to just write about them yourself. And the critiques of the field are two. Forget my olfactory story. Take any intergenerational inheritance phenotype out there. How is any of that information about the environment getting to the sperm or the egg? In 1938, August Weismann proposed that the sperm and egg are immune to everything around them, all insults around them. So how is any information getting to the sperm and the egg? And I'll address that in a minute. The critique B is this concept of epigenetic reprogramming. So you have epigenetic marks being laid down, histones, DNA methylation, whatever you want, but they get scrubbed away. They get erased at different points during development. So how is any marks which are laid down by environmental experiences, how are any marks escaping that epigenetic reprogramming? That's a bigger question than me, but thankfully we have a ton of people who've been working on this stuff, and most recently Azim Surani put out a paper, a companion piece in a pretty big journal, where they looked at primordial germ cells, human primordial germ cells, and found that there were 25% of the loci that escaped reprogramming. These 25% of the loci were associated with obesity and schizophrenia. Now, what the logic behind that escape from reprogramming is, why and what ramifications that has, I think, is the wave of the future. But at least we know that there are genetic loci that do escape epigenetic reprogramming. So the question is, how is this happening? When we try to put out this paper, and I think it's been a while, but in the discussion, we said RNA might be mediators of intergenerational inheritance. I can show you the email where the editor or the reviewer said that is the stuff of science fiction. 2017, that's exactly what we know. All right? All of these papers, in some shape or form, have done this experiment. They've conditioned their animals, or they've stressed their animals, or they've manipulated the diet of their animals. They've gone and extracted sperm. They've got an RNA from that sperm. They've injected that RNA into single-cell zygotes, and they've recapitulated all of their effects. Tracy Bale's group took 9 microRNAs and injected it into single-cell zygotes and got all of their effects. So at this point, RNA are the front runners in terms of carrying intergenerational information about ancestral environments. And so what we have working to our advantage right now is we have a point of trauma, of stress. We have the nose. So what we're doing right now is we're profiling RNA in the main olfactory epithelium in animals that were stressed versus not with the odor. We're looking at circulating exosomes, which are these tiny vesicles coursing through circulation. And then we're looking at sperm to see, is there any correspondence? And just to give you, I just find it amazing that people have thought about this stuff before. Way back when Charles Darwin proposed this concept called gemmules, little gems floating through circulation that would allow for information to go from one part of the body to another, including the germ cells. We would call those exosomes right now. Exosomes blebbing off from every tissue that we can conceivably think of, what those are carrying, where they're going. I think that's the stuff that I know a lot of people in the room are working on. If you are engineering exosomes that I can then track, please, please, please drop me an email. We would love to be able to do that. I've had some conversations with people, but I want to further those conversations. So in the last minute or two, I want to tell you, where is this going? So when I send this stuff to NIH, they tell me that olfaction is too niche. It doesn't have translational relevance. So hopefully NSF will bite, right? We can hope and pray for a miracle. But truth be told, I've worked on a lot of different species, but I wanted to come back to the psychiatric space. And so leveraging really generous collaborations at the Yerkes Primate Center, we're doing some stuff in non-human primates. So I'm just going to show you what we're doing, and the data are just coming off the sequencer, and we're doing the bioinformatics on it. So what we're doing in collaboration with people like Mars Sanchez and Elise Morin, Mark Wilson, Vas Mikopoulos, Jessica Raper, and Jocelyn Bacavelier, is we have, depending on males or females, we're getting plasma exosomes or sperm from animals who've been maltreated as infants, from individuals who are of a high or a low social rank, from individuals who've been raised by their mothers versus raised in the nursery, and we also have individuals who are administering cocaine, self-administering cocaine. And so the question is, are there biological signatures that are conserved across these different stressors within the blood and in the sperm that will then predict what future offspring, or even within a generation, will predict what the behavioral phenotype will be? So here I'm showing you our ability to isolate exosomes from the mouse as well as the monkey. And then we have RNA coming off, really small RNA coming off these particular exosomes. You don't expect RNA, long RNA species to be found in these exosomes. And then here I'm showing you our ability to get really high quality RNA from macaque sperm, and we do this in the mouse sperm all the time. So why bother? What's the bigger goal? Once we identify these signatures, we can either engineer those signatures in the lab and then inject them into embryos. But what we're right now doing, and this is the work that Haj Awed is really pioneering in the lab with the help of Dr. Anthony Chan, who's helping him tremendously, we're injecting stressed RNA, which we obtain from sperm of those animals who've been maltreated or not, on cocaine or not. And then the idea is to inject that stress RNA into the male pronucleus, let the embryos develop to the 16-cell stage, and that embryo now has a legacy of infant maltreatment, of maltreatment and cocaine in some cases. And the idea is, how are we going to push or pull the embryo down a risk versus a resilient trajectory? And so that's, I think, everything that I wanted to talk to you about today. These are the many faces of us. Hasa and Elise are not in the lab, but really are helping us with the bioinformatics and a lot of the monkey work. I haven't had time to talk to you about Archana's work, where we are working on fear within a generation and the neural circuits underlying fear inhibition. Christy's been working on astrocytes, and all the undergraduates who help out with their own projects. Thanks to my funding sources. I find that there's too much shade thrown on too many people's work these days. I don't believe this and I don't believe that. I'm not in the religion business. So this is my antidote to that. These are people whose work inspires and informs me. And thank you for your attention. Yeah.
Really great talk.
Thank you.
I think looking at the fear induction and extinction in the F0 generation is really interesting and gets at more of the mechanism. But what I think you really want to know is, what if you extinguish
F1s?
Yeah,
right. Yeah. So you bring up a great point. The ship has sometimes already sailed with the F0s, and so how do you go in and reverse that in a generation that you have access to? And so, absolutely, that's the kind of stuff that we need to do. Yeah, I agree. Yeah.
As a follow-up, maybe I missed this in the different conditions that were there, but the sensitization with ACE was done at the F0 generation, and the extinction was done on the F1.
In the F0s as well. In the F0s as well.
The extinction was done in the
F0s, right.
Does extinction by itself without the pre-sensitization have any effect?
It doesn't. Right, so that was our ACE exposure group, and we don't get that effect. So I think there's something about the stressor that's allowing for this effect to go through, and we don't know what that stress phenotype is. This one, yeah.
So the mechanisms in the F0 that increase the amount of neurons that express the receptor, do you think those mechanisms are the same as what's being used in the F1 to increase the amount of neurons in that as well? Or do you think
In the F2, you mean?
the F1 and the F2?
Yeah.
Completely different mechanisms?
So that's a really good question. I don't know the answer to that. People talk about genetic assimilation in some way. So you have maybe an epigenetic mechanism, and then it gets fixed in the genome. I think that's way too early for something to get fixed. So the hypothesis I have is that if we actually took F3, F4, it would actually get diluted away, unless you give it a booster, so to speak, because it doesn't make sense for an animal to remember this particular salient environment if acetophenone is no longer around. And so that's something that we're going to test in the lab to see, do you need an environment to constantly be salient to get this propagation across many generations? And Go ahead.
And so BPTF and NERP, I think, is involved in the specification of the olfactory neurons.
Tons of different, yeah. And so, it's a question of which ones do we look at? Is it going to be only one versus another? So you bring up a point which I think is well taken. The olfactory field typically thinks of odorant receptor choice as being stochastic or random. And so then it's a question of, are we really forcing olfactory choice with some of these kinds of protocols that we're doing? And I think the jury's still out on that. And you'll have people who say, yes, there can be activity-dependent olfactory receptor choice, but then a lot of that is also stochastic. So Philomen is we're trying to put out a paper where it's more survival, not so much neurogenesis, not so much proliferation. So we've done some BRDU, EDU studies where it's more survival, not proliferation. Yeah.
Was it only the male mice that had the treatment with the foot shot sensitization?
It goes through the female lineage too. The reason we focus on the male mice is just because we can get a lot more sperm compared to eggs.
So if both parents have had this sensitization, is there like an increased effect or is it the same?
Have not done that experiment, and I don't know what we'd expect. There's a lot of literature to suggest that mom can buffer things that happen to dad, and so I don't know, that could go either way. Yeah.
Great talk, Brian.
Thanks.
Just a quick question. How many generations, so for example, predatory responses
Yes.
are innate,
Yes.
right? So, but at the very beginning,
Yes.
maybe they weren't.
Yes.
How many generations do you think would have to go through to make it innate?
Right.
And have you ever thought of using a predatory response as like a control to say F0, F1, F2, it always shows the same?
Right. So I think so the first part, yes, a lot of people have talked about the fact that how did fox urine or cat odor become an innate predatory scent, and at some point somewhere it needs to have started that initiation for being hardwired. And so is this that kind of plausibility? So Gene Robinson, who does a lot of work with honeybees, wrote a piece on the evolution of instinct and cited this work. I stay away from those kinds of things because they're out of my wheelhouse, but that premise has been put out there. Using it as a control, I think I see your point, but I feel like fox urine or cat odor is going to be so innately aversive. These effects are more subtle, and so I think then we're comparing different shades of responses, and so I'm not so sure what we'd be able to tease apart. I'll go there and then come to Sam. Yeah. Did you have? Yeah.
I had a question. So maybe you may cover this, but have you looked at rewards instead of fear responses?
I did not cover that, so thank you for asking me. I'm only a consultant on these projects. I'm not involved. Regina Sullivan does a lot of work where she can pair an odor with mum, and that becomes in rats, and that becomes an appetitive odor, and there's a preference response. So when Regina and her students pair a particular odor with mum, they find that the preference goes to the F2 generation. And then they're trying to do some studies with respect to the mechanism as to how that's happening. So we don't think it's only an aversive experience. We think it's appetitive as well. Yeah. I'll go there and then come back. I'm just spreading the wealth.
So is there a continuum of, in the sperm, severity of predisposition to aversive behavior as a function of severity of the initial condition?
Right. So I can't answer that question. I guess let me try and answer it in different ways, maybe, but I don't think this is an answer. One is we can say which males learned about that odor a lot more, the ACE plus shock association, and which gave us the highest fear response, and then do we see correlations with the anatomy and the behavior in the offspring. We haven't done that. We don't select any males at all. So we don't pre-select. We just take all the males and all their offspring. So that's one way. The second way, I think this is where what you might be getting to is I don't know the answer in terms of are all the 3 million sperm in ejaculate affected equally with an environmental experience, and that's going to have single cell approaches that we're going to have to take for that, and also a question of which sperm precursors. Now I'll preempt the question about eggs. I don't know whether all eggs are going to be affected. You have eggs that are arrested in a particular state of the cell cycle. So then does it matter if mom is only influenced, only gets an environmental trauma when she's just ovulated and there's one egg which will bear that imprint, or whether all her eggs bear that imprint? Those are questions that I just don't know the answer to. I'll come back here.
So I was really impressed that the morphological signature and effects were present in the primary sort of olfactory sensory circuit,
Mhm.
like pretty much right at the beginning.
Yeah.
The sensory systems share some, all the different sensory systems share some general mechanisms, but also have very unique specialties to them. I sort of see why you used odor
Right.
here, but is there data, or could you at least speculate on whether you think these same kind of mechanisms might be manifest in the same ways in other sensory conditions?
Sure. So let me just take a step back. Your question is a nuanced one. In the F0s, I don't think the enhanced structural representation is related to the behavior, because you can train an animal to acetophenol plus shock and get a behavioral response tomorrow, but the glomerulus response comes on 3 weeks later. And so there I think there's a dissociation. We went with olfaction because we knew that this genetic architecture existed. So in thinking about could this be done with auditory cues, I suspect not. But I can envision it being with visual and with gustation, because there are similar mechanisms in play with rods and cones being encoded and representations being changed based on environments. So I can see visual and gustation, but not so much auditory information. Hebla.
Just to try and tie it back to translational relevance. How strong are the effects with single exposure compared to multiple exposure?
Yeah, I mean, I do not think we'll get this effect with single exposure. We haven't tried this this experiment, but I think that's a good point, because when you talk about something that is traumatic, it's not you talk about a multiple hit hypothesis. You're talking about early childhood abuse, and then you have something happen in adolescence, and then something happen in adulthood. And so there are these different phases of vulnerability where I don't think it's only a single exposure that's going to give us an effect. Or even within, if early childhood abuse was the only trauma, it needs to have happened for a sustained period of time to allow for those effects to actually manifest. And so I think the trauma or the stress in our animals, the stress needs to occur repeatedly for us to get some effects. Yeah.
Is there any program to take sperm samples from men before they go into the army?
Yeah. So I'm talking to someone to try and figure that out, to see out in North Carolina, to see if we can get sperm from soldiers who before they go into the army versus go into combat versus after they come back. But that stuff comes with its own attendant issues, but we're trying to do that. Yeah. All right. Thanks, everyone.